Cellular respiration is the process of breaking up organic molecules with the help of oxygen in order to extract energy out of those molecules and use that energy to make ATP, which will fuel all the endergonic reactions in our bodies. Now, how this happens is a very detailed process, but all organisms respire. So all organisms go through the process of cellular respiration in some way or another. Organisms that produce their own organic molecules through photosynthesis or chemosynthesis, so they make organic molecules out of inorganic molecules by building up small inorganic molecules into larger, more complex organic ones, and getting energy to do this from either the sun or the geothermal activity from the inside of the earth. Then these autotrophs, after building up their own organic molecules, they will actually break down those organic molecules they made in order to make ATP. Whereas heterotrophs are organisms that cannot produce their own organic molecules from inorganic ones. So they have to eat the autotrophs in order to get the organic molecules that they made, and then break up those organic molecules to get energy to make ATP. So all organisms, plants, animals, fungi, protists, bacteria, archaea, they all use cellular respiration in some way to extract energy from organic molecules. The difference is whether they made the organic molecules themselves or not, is the difference between autotrophs and heterotrophs. So in summary, photosynthesizing organisms like plants, they take energy from sunlight and they use that energy in order to build sugars from very small and simple inorganic molecules like carbon dioxide and water. So the process of building up glucose in this example is an endergonic process. It requires input from the sun because we're taking simpler, more stable molecules like carbon dioxide and water, and we're trying to rearrange those atoms into higher energy levels to build up a more complex, more bigger in size and less stable glucose. Okay, so photosynthesis is an endergonic process of building up glucose. Then the plant cell itself will utilize the energy that originally came from the sun that is now stored as potential energy in the bonds of the glucose, and we will break up that glucose with the help of oxygen. That reaction is, where's my little pointer? This reaction is an exergonic reaction because we're going to take the glucose and we're going to break it up into simpler and more stable molecules. That energy that we're going to extract through this process is going to be used to make ATP. And we'll be left over with these simple, stable, low energy molecules, carbon dioxide and water. And in order to put them back into something more complex, we need to get energy. Again, usually from the sun, through photosynthesis, which is an endergonic process. Then we make glucose, we break it up, which is an exergonic process, in order to build up ATP, and so on and so forth. The more complex the more organized and the larger an organism is, the more energy it requires for maintenance. Okay, so there's some averages of kilocalories consumed for a person that is about 67 kilograms. So what's a kilocalorie? Of course, that's 1,000 calories. And what's a calorie? 1 calorie is the amount of energy needed to heat up 1 milliliter of water by 1 degree Celsius. On food labels in the United States, and one of the students pointed out, especially for human food, as opposed to like pet food, instead of writing kilocalories, for some reason, American food companies decided they're going to just write calorie with the big C and have that mean the same thing as kilocalorie. So on food labels, if your chips say that one serving is 300 calories, that's actually 300 kilocalories. It's just an odd, confusing thing on food labels. All right, so this process of breaking down organic food molecules in order to get energy out for the creation of ATP, this process is done in a series of many steps. And every single step is a version of electrons being broken off and moved somewhere else. And the electrons that are broken off carry with them energy. You know that electrons are bouncing around in those electron clouds depending on what energy shell the electrons are in. Like the further away they are from the nucleus, the more energy they have. And in chemistry, the loss of electrons is called oxidation. A related process, dehydrogenation, is when you lose electrons, but it's also accompanied by a proton for stability. Remember that hydrogen is 1 electron and 1 proton. So dehydrogenation is the loss of electron and a proton. Now, in order to break off the electrons, those electrons that are lost by oxidation or dehydrogenation, we'll just call them all oxidations, even when those electrons are accompanied by hydrogens, because it's still under the overall umbrella of oxidation and it's easier to address it that way. Every time electrons are lost or broken off by oxidation, those electrons have to go somewhere and get attached to something else. So another molecule will gain them. And the specific chemistry term for gaining of those electrons is called reduction. So in order to remember which term is used for loss of electrons versus gain of electrons, there's a mnemonic device. Leo the lion says grr. Lose electrons is oxidation. Gain of electrons or gaining of electrons is reduction. Okay. Since the two reactions, oxidation and reduction, generally go together, we call them redox for reduction and oxidation, right? So electrons are broken off from one molecule and they are reattached or gained by another molecule. Together, those two reactions as a pair are called redox. Okay. One specific type of molecule that exists in your cells all the time, called NAD+, stands for nicotinamide adenosine dinucleotide. It's a type of modified RNA nucleotide. You don't need to know the full name, but it's NAD+ is what you will know. It is a molecule that helps take electrons off of something and move them somewhere else. And the way it does that is it grabs 2 electrons and 1 proton when it helps electrons come off of organic molecules. And then when it has been fully loaded with those 2 electrons and 1 protons, it is now called NADH. So when you see it as NADH, it means it is loaded with 2 electrons and a proton, but we don't care about the proton. When you see the name as NAD+, it means it is empty, or it's not carrying 2 electrons. The reaction is reversible, so we call this molecule an electron carrier. It's like a shuttle for electrons. So for example, here's NAD+, it's just represented by this blob. And here is some kind of a food molecule, organic molecule that is energy rich. And this transaction is going to be catalyzed by an enzyme. So an enzyme will help speed things up here. It's a very specific enzyme for every possible case. So in our generic example on this slide, an enzyme will bind to its active site, NAD+, so this empty molecule. It's not empty, it's made up of atoms, but it's not full of extra electrons. And it will bind this food molecule. The food molecule is made up of a bunch of different atoms, but this diagram is not specifying that because it doesn't matter. Then 2 electrons and 1 proton will come off of the food molecule and attach to NAD+, which will now make it NADH. The food molecule, which we can now call the product, has lost 2 electrons and a proton and will be released from the active site of the enzyme. NADH will now take those electrons and the proton and take them somewhere else. So overall, this whole process that we're about to learn of cellular respiration is dozens of these redox reactions, meaning some kind of a electron carrying helper molecule is going to grab electrons off of an organic molecule, so off of food type molecule, and it'll carry them somewhere else. So dozens of these redox reactions that are assisted by molecules like NAD+ or other electron carriers. All in all, the goal is to grab electrons when they are in a higher energy level and move them to a different molecule or to a different environment where they will be stable at a lower energy level. The net difference in energy can be released and harvested for making of ATP. The types of respiration in our cells differs depending on what molecule is there at the end to grab those electrons after we've taken all the possible energy out of them. If that final electron acceptor is oxygen, then we say that it is aerobic respiration, aerobic. If the final electron acceptor is not oxygen, but some other inorganic molecule, then we say it is anaerobic respiration. And if the final electron acceptor at the end is some kind of organic molecule, then the process is called fermentation. This will make much more sense when we can go through the process as an example or several examples, see how it happens, then you can circle back to these terms and see if you can remember these definitions in context. In general, aerobic respiration, which is this process of harvesting electron energy from organic molecules we get from food, right, in a step-by-step manner, if it happens in the presence of oxygen in your cells, then it resembles like combustion or burning of other organic things, other organic molecules. Notice here we're starting out with some carbohydrate, and in the presence of oxygen, this larger, more complex, more organized molecule that has more potential energy stored in it, in its bonds, will be rearranged to create smaller, lower energy, but more stable molecules. And the net difference in energy will be released. So this is an exergonic reaction. Notice you started with one big carbon and 6 oxygens, and now you have 6 carbon dioxides and 6 waters, 12 molecules. So you definitely went from more organized and larger to less organized, smaller molecules. Higher total potential energy stored in the bonds. Here's lower total potential energy stored in the bonds. And the net difference in energy will be released and used to make ATP. If we take a carbohydrate and just light it all on fire at the same time in the presence of oxygen, like you would a piece of wood, which is also made of carbohydrates, so this is, or gasoline, which is also not a carbohydrate, but it is a hydrocarbon, so it'd be a similar process. All the energy that was released would be released in the form of heat and light, and those are not useful for our cells. So we will do this overall thing, but we're going to do this in a different way that is much more controlled, much smaller incremental steps, and all of that is to reduce the amount of heat that's produced because it's wasteful. So the large amount of energy that's in the carbohydrates must be released in small steps rather than all at once. Here it's a kind of cartoon diagram of how we take electrons that are in the covalent bonds of the food that you eat, like glucose. Those electrons are in higher energy shells, and so they have higher energy. We're going to somehow rearrange everything so that the electrons can fall to a lower energy level in a different environment, and then do it again and again. And each time, as it goes to a lower energy shell, the net difference in energy can be released and used to make ATP. And because it's done in such small controlled steps, where electrons grow from having a lot of energy to almost no energy, we are going to be able to create that ATP. One more word about the electron carriers. The example we gave you is NAD+, and let's look at the structure of that in a second. But in addition to having NAD+, there are other electron carriers that do the same thing. They're molecules that are already in your cell that are not made or permanently consumed during these reactions. It's just an assistant that's always there to help with the process of respiration. They easily grab some electrons, sometimes electrons and protons, and then they can easily release them in a different environment. Once they release them, they return to their original shape and do the process again. In the case of NAD+, the molecule is made of ribose sugars, 2 ribose sugars, adenine, nitrogenous base, phosphates, and then there's like a different group up here. So it's a modified RNA nucleotide or nucleotides. It can attach electrons and a proton right here to this carbon. So when NAD+ is reduced, remember, gain electrons is reduction. When it's reduced, it is now called NADH, and that means it has grabbed high energy electrons, and it is probably swimming somewhere in your cell to release those high energy electrons. And when it does so, it will lose high energy electrons, therefore it will now become oxidized NAD+. So when you see it as NAD+, it's the oxidized version of this carrier. meaning it is not loaded with those electrons. When you see it as NADH, it does have extra electrons loaded, and it is the reduced version of this electron carrier. So now let's go ahead and get started with the specific process, the actual overall process of cellular respiration. In our example, it will be in eukaryotic cells. And we're going to look at how we get energy from glucose. If you're thinking, well, how does glucose get in there anyway? You eat food. Let's say I just ate some mashed potatoes. And those mashed potatoes are largely made of starch. The starch will go into my stomach, and in the stomach, it will start getting broken down into individual glucose monomers. Remember that starch is a polysaccharide, which is made of covalently bonded glucoses in a long chain. In the stomach, we have enzymes that help speed up the breaking of starch into individual glucose monosaccharides. Then this slushy mixture from our stomach will move to the small intestine. And from the small intestine, everything that's already been broken down to monomers, so glucose and amino acids and individual monomers of all kinds, they're gonna seep through the wall of the small intestine. And across the wall of the small intestine is blood vessels. So glucose that used to be a part of that potato will seep into the blood stream from the small intestine. And now you have a bunch of glucose in your blood vessels. Well, your blood vessels go around all of your tissues. And any tissue that needs energy, whether it's a liver cell that needs energy, or muscle cells in your legs that need the energy because you're walking, or it's your brain cells that need energy, then those cells can grab the glucose out of the bloodstream. It's almost like an ice cream truck driving by through your neighborhood. If you need ice cream, you run out and you grab the ice cream and take it into your house, right? So glucose is just kind of being swimming around your bloodstream. If a specific cell needs glucose, then it will grab it as long as there is insulin, which is a hormone. It's a protein that's also a hormone in your bloodstream. That insulin will help glucose that came from your food enter cells of specific tissues. So now, let's say it's a muscle cell that has taken in glucose with the help of insulin. Now there is glucose inside of your muscle cell. Well, glucose cannot be directly used to fuel endergonic reactions in your cell. So if you need energy for active transport, to move vesicles from rough ER to Golgi, glucose cannot help with that directly. Glucose has energy stored in it, kind of like a check, a personal check has money in it, but you can't just take a check that, like, let's say your friend wrote you a check because they bought something from you for $50. You can't take that check and go to the grocery store and say, here's $50 to pay for my groceries. That can't be used directly. You have to go to a bank or do it online and exchange your check, deposit it, and then take out $50 in cash. Then the cash can be used in a grocery store. In the same way, glucose is in your cell, and so technically there is potential energy to fuel things in your cell, but it just cannot be used in that form. So the energy that is stored in the covalent bonds of your glucose needs to be converted into energy that's stored in ATP. And then ATP is like cash. It can be used to power just about any endergonic reaction in your cells. So how do we convert the energy that is in the covalent bonds of the glucose into energy that's in the covalent bonds of ATP? And that's the specific process of cellular respiration that we are studying right now. It happens in 4 main stages called glycolysis, pyruvate oxidation, Krebs cycle, which is also called citric acid cycle, and oxidative phosphorylation, which has 2 subcategories, electron transport chain and chemiosmosis. The first process, glycolysis, happens in the cytoplasm, while all the other processes happen in the mitochondria of your cells. Okay. So let's go ahead and get started. Let's remember the context. You ate a bunch of food, including carbohydrates, and through the process of digestion, a bunch of glucose has made it into your cells. Now we need to convert the energy of that glucose into ATP potential energy. What's the first step if we are starting with glucose? The general first step is called glycolysis, but it is itself made up of many steps. As you can see from the word glycolysis, it's the breaking up of glucose. So we're going to take the glucose and we're going to break it apart. Specifically, we'll take the glucose, which as you remember is made up of 6 carbons, and we're going to break it exactly in half to create 2 molecules that have 3 carbons each. So if you imagine having one big thing that's made out of 6 carbons and you break it in half, you now have 2 molecules that are 3 carbons each. Those 2 molecules are each called pyruvate. So overall, glycolysis will be taking a glucose and breaking it in half, resulting in 2 pyruvates. But this will happen in a 10-step biochemical process, which occurs in the cytoplasm. And from this rearrangement, from breaking up of glucose and rearranging the atoms to create pyruvates, we will net gain some useful energy molecules. Let's see how that happens. I'm going to have to show you a diagram of how this happens. Glucose is a rather stable molecule. Remember, it has to go from your stomach to the intestine, to the blood, to the cells. And the whole time, we don't want it to be really unstable and reactive because that could cause problems. So it's a stable molecule that's good for moving around. In the same way that personal checks are good and stable, because if you lose it, no one can use that money, right? I don't know, checks are pretty stable, unlike cash that can be lost and stolen and used directly by other people. I don't know if the analogy holds. But anyway, glucose is pretty stable. So in order to break it in half, we have to invest quite a bit of activation energy to shake up those covalent bonds that make up the glucose and break them and rearrange the atoms to become pyruvates. So it's like you've built something that's pretty stable, and in order to now take it apart, we need to invest some energy. And the energy that we invest is 2 ATPs. So let's back up. glucose has entered your cell, and we're going to try to rearrange the atoms in the glucose to extract some energy. In order to rearrange the atoms, we need to break up the existing structure, but it's pretty stable. So to break it up, we have to kind of use little energy from one ATP and another ATP that's going to kind of like little bombs going off next to the glucose, and it's shaking it up and breaking it apart. So far, we've actually lost energy, right? 2 ATPs had to be invested. But once that happens, there is like a quick domino effect of rearrangement. Each step requires a bunch of enzymes to help it. And the electrons that were holding the 2 halves of glucose together are going to get loaded onto NAD+, making it NADH. And these NADH molecules are going to swim away to the mitochondria and used in a way that I'll show you later. Enough energy is released from the breaking up of glucose to put together 4 ATPs. Okay, so we invested 2, we made 4 ATPs, so that's a net gain of 2 ATPs. And in addition, 2 NADHs have been loaded up with high energy electrons. And that energy from those electrons is going to be used in a later step. Electron transport chain is a series of membrane-bound electron carriers. And actually, I'm going to show you the picture of it. Okay, so these blobs, blue blobs, are representing protein complexes. So actually, multiple proteins kind of stuck together, working together, that are spanning through the membrane of mitochondria, the inner mitochondrial membrane. The reason why there are all those folds in the inner mitochondrial membrane is because we want more surface area to have many, many copies of the same arrangement. Many, many copies. So you can do this process at the same time on multiple stations, so it can be fast and efficient. Okay, so there are these 3 protein complexes, and there's going to be a 4th one as well, just a little further down the membrane. And let's see what they are for and what happens. So NADH, remember this is the reduced version, the one that's gained electrons. So this is the electron carrier molecule that has attached to itself 2 high energy electrons and a proton. And it's going to come and drop those high energy electrons into this protein complex. We call it protein complex number 1 because it's the first one. The proton is going to break apart and stay in the matrix. Now NAD+ is back to its oxidized form, and it swims back and helps with glycolysis or pyruvate oxidation or the citric acid cycle again. So goodbye NAD+. The energy in the electrons is going to be used by this protein complex to drive active transport of this H+. It's an unusual way to power active transport that only happens here in the mitochondrial inner membrane. Energy from the electrons that have just been dumped in here will be used to drive active transport of H+ against its gradient from the matrix into the intermembrane space. Now the electrons still have some energy and they'll be shuttled to the next complex, complex 2. Again, energy from these 2 electrons is going to be used to pump yet another proton against its gradient from the matrix to the inner mitochondrial inner membrane space. Those electrons are then now much lower energy and they will come to the 3rd protein complex. And again, the very, very last little bit of energy from those electrons will be used to pump proton against its gradient from matrix to the inner membrane space. So far, the energy that was in the high energy electrons of the glucose, which was shuttled onto NADH and now dumped to this electron transport chain, that energy so far has been used to drive active transport to pump protons against their gradient. By the time the electrons make it to this 3rd protein complex, they are so low in energy. They're barely moving. They're just barely vibrating. And they don't even have enough energy. Imagine the electrons bouncing, and now they're just barely moving. They're so low in energy. They can't bounce out of that protein. However, we have access, we as animals or plants or fungi or protists, we have access to a very electron-negative molecule. What is the most electron-negative atom that we know of that exists in our bodies? Is oxygen. Oxygen is going to be hanging out here in the mitochondria, and because it's so electronegative, it is able to pull those electrons that now have almost no energy left in them. It can pull it out of complex 3 of this 3rd protein and combine it with the oxygen protons and make water, which will be waste. The reason why it's important to pull those electrons out is because there's only one spot for electrons. And if we want a new NADH to dump its electrons in, we need space that is empty in each of these complexes. So oxygen pulls it out. Now there's room for more electrons to go through, which means there's energy to pump more protons against their gradient. FADH2, which is another electron carrier that we met at the very end of the citric acid cycle, it also dumps its 2 electrons, but it bypasses the first complex and puts them here in between 1 and 2. Therefore, the electrons from FADH2 are able to only power the active transport of 2 protons, whereas electrons from NADH are able to power the active transport of 3 protons. But other than that, the electrons that come from either of these electron carriers are doing the same thing. So far, we have collected high energy electrons from glycolysis, pyruvate oxidation, and the Krebs cycle, all for the purpose of pumping protons against their gradient, and therefore establishing high concentration of protons in the inner membrane space in comparison to the matrix. We have made a very strong gradient by actively pumping protons against their gradient. And the reason why we did that, why it's going to be useful, is because now there's this really strong physical desire of these protons to go across the membrane down their own gradient. They, of course, can't, because ions can't get through the membrane, right? You know that they cannot fit through the membrane. And there's only one opening for them. And that is this fourth protein complex, which is called ATP synthase. You should know the name of this because it's very descriptive. ATP synthase. We have huge proton gradient. It's not only a concentration gradient, it's also an electrical gradient. Lots of positive charges here, not very many positive charges on this side of the membrane. And then a concentration gradient, lots of protons in the inner membrane space, fewer on the other side. We have created a huge gradient, and the protons can't go through except for this hole that is in the ATP synthase. This is kind of like facilitated diffusion. There is a spot for ions to travel down their gradient. So they gush through. The stronger the gradient, meaning the bigger the difference between the two sides, the more they gush through with a faster chutzpah. And as they gush through, it causes the rotor and the stalk to rotate, physically rotate. This protein is crazy. It's like a merry-go-round. And as it rotates, it provides mechanical energy to put together ADP and phosphate into a functional, fully formed ATP. This is the same way as in the olden days, you had windmills that as the wind was pushing the windmill and the windmill was rotating, it was able to use that energy to grind wheat, right? Because there would be these special mechanical things that were attached to the rotating propeller-looking things, and it would grind wheat. Or a water turbine, that as a river was flowing through a water turbine, as it spun, it can create electricity that can be used for something, right? Or how modern windmills work. But instead of the rotation providing electricity or grinding wheat, in this case, the rotation energy is directly used to put together ADP and phosphate to create ATP. So the whole reason why we were saving up those high energy electrons was so that we can create a strong proton gradient. And the reason we wanted a strong proton gradient is so that the protons could then be gushing through the ATP synthase, causing the rotation, and that energy would be used to put together ATP. Of course, if we are not constantly pumping protons against their gradient, then eventually, within just a couple of seconds, all the protons would gush down their concentration gradient and form equilibrium. And then there would be no more gushing, right? This is a process that's extremely complicated, and takes time to wrap your brain around and go through so it makes sense. I worked in my graduate studies, I worked with several chemicals that interfered with various steps in this process, so that once we interfere with one step, we can see how everything works. So for example, there is a chemical called rotenone, and it blocks the first protein complex, so no high energy electrons can be going in there, and therefore no pumping across it can make sense. It can happen. So NADH cannot drop anything off there, and therefore there is no place for NADH to drop electrons at all, because it can only do it at complex 1. Therefore, we really slow down the whole pumping process, because only FADH2 was able to contribute high energy electrons, because it bypasses the first protein complex. You've probably heard of cyanide as a poison, and of course of carbon monoxide as a poison. The reason why both of those molecules are deadly is because they completely stunt, that's not the right word, they clog complex 3, or that third protein in the electron transport chain. And because of that, the whole thing gets backed up and doesn't work. And so a gradient cannot be created, a proton gradient can't be created, and the gushing can't happen, and so ATP can't be made. And when you don't have ATP, you die. Right? Also, oligomyosin blocks the ATP synthase. So again, ATP can't be made if this chemical exists. Interestingly, DNP is a class of molecules that were used in a pill form in order to help housewives, mostly in the 50s, to lose weight. I want you to think through that. The way that this molecule worked is when a person eats it, it goes all the way to your mitochondria, and the molecule literally creates a big hole in the inner membrane of the mitochondria. Just a hole, just an uncontrolled pore. And the reason why that's problematic is now protons were going through those holes, and therefore there wasn't much of a gradient. This made ATP synthesis very inefficient in this whole process, is because you had leakage through the inner membrane. Okay? Can you think of why that would cause someone to lose weight? It worked pretty well. The reason why they stopped prescribing these meds is because there were a bunch of deaths that occurred due to this medication, I guess. Unfortunately, now DNP is sold as a quote supplement, and it's sold online in various forms. And when I paid closer attention to this, almost every year I saw somewhere in the news that a college student somewhere, usually a male college student, died because of taking DNPs as a quote supplement for reducing body fat and increasing muscle mass. It's extremely dangerous. I advise you with in every way I can, to never use these supplements. They're extremely dangerous, especially when they're just purchased online. You don't know what's in there and how they will affect your specific body. Okay, so the word chemiosmosis is the combination of electron transport chain and the ATP synthase, which is also called oxidative phosphorylation. And it works because the accumulation of protons in the inner membrane space drives protons into the matrix via just passive, regular diffusion, as if it was facilitated diffusion. In fact, it is facilitated diffusion through the ATP synthase. That ATP synthase is the only spot through which those protons can gush through to attempt to form equilibrium, because the membrane itself, the phospholipid membrane, is relatively impermeable to ions. So most of those protons can only re-enter the matrix with the goal of creating equilibrium, so to speak, through that ATP synthase opening. And that will cause the ATP synthase stalk to spin, and the spinning energy makes ATP form from ADP and phosphate. The summary of this whole process, glycolysis, happens in the cytoplasm, pyruvate oxidation, Krebs cycle, electron transport chain, and chemiosmosis, all is in this diagram. Why is it that if we do not have oxygen, so if a person is not breathing, they die? Why is that? When there's no oxygen to pull the electrons out of complex 3, those electrons that are now very, very low in energy, to pull it through complex 3, then it's like all the bases are loaded, like in baseball. There's already electrons stuck in here, electrons stuck in here, and electrons stuck in here. And that means that new movement of electrons is not possible. And it is only when electrons are moving through that we can pump protons. So if oxygen is not pulling the last electrons, no more electron flow is happening, so no gradient can be formed, and therefore protons will not gush through, and therefore ATP will not be made. Okay? So notice that the NADH we made during glycolysis in that first step, it immediately went over here. And so did all of the electron carriers. We just didn't follow them right away as we were learning this information. All in all, during glycolysis, we created a net 2 ATPs, and then the NADHs we used for glycolysis, those high energy electrons that were loaded on here from the glucose, made about 6 ATPs when they went to the electron transport chain and ATP synthase. And you can see how kind of all the calculation is here. So in general, from one glucose, we're able to make about 36 to 38 ATPs. Only 2 of them are done in glycolysis, and 2 more are done directly in the Krebs cycle. That means that the electron carriers that carried the high energy electrons were the ones that allowed for most of the ATP in your body to be made. So it's those high energy electrons that ultimately provided energy to put together ATP. And then we use all of these ATPs to power all the endergonic reactions in our body. This is a cool diagram that shows that sometimes the products of these processes can inhibit or shut off the flow of the biochemical pathways. So if there is a lot of ADP around in your cells and in your mitochondria, that tells your cell that we need more of this process to happen because we need this ADP to become ATP. So it activates everything, turns on the process. These words right here are names of enzymes, the names of intermediates of the process. Remember, we started with glucose, we ended with pyruvate, but these are the names of the in-between molecules that we did not name. If you have a ton of ATP already, your cell is like, we have lots of ATP already, we don't need any more. Then the high concentration of ATP, by just a lot of them bumping around and being in your cell, it will inhibit or shut off glycolysis, because your cell doesn't need to be working hard going through this whole process if you don't need any more ATPs. Let's say you were running and then you stopped running and your cell already has a lot of ATP and your energy need is now very much lower than it was before. So it makes sense to shut off this process and let your body rest. Similarly, if you have a lot of NADHs, which means there are already a lot of high energy electrons in your mitochondria, you don't need to load more, then it will inhibit as the pyruvate oxidation step because we already have a lot. We don't need any more. Okay, so you do not need to memorize what inhibits where and how, but I would like you to be aware of the ability to regulate this process. So if you already have a lot of the products or even the NADH, which will become useful in creating product, then you don't need to keep running this whole operation. And if you have a low amount of the product you need, which is recognized as a high ADP concentration, then it activates and turns on this whole process. It's pretty cool, right? So that was the process of breaking up a glucose molecule to get energy to make ATP. And that ATP is going to be used to drive endergonic reactions. At the very end there, when I showed you the electron transport chain and ATP synthase, that process was making of ATP by oxidative phosphorylation. That means you used oxygen to phosphorylate something. To phosphorylate, it means to add a phosphate group, right? You had ADP and you had to add a phosphate to it covalently to create ATP. So that's phosphorylation. Oxidative phosphorylation, it's in the presence of oxygen. ATP synthase will use energy from that proton gradient to put together ATP from ADP and phosphate. Okay. But when we made ATP during glycolysis, you were not using the same process. So think back to glycolysis. You were not using the same process because we did not have a gradient. We were not even in the mitochondria. And we certainly were not using an electron transport chain. And that's when you have transfer of phosphate group directly onto ADP, just kind of having enough energy to smash them together. And we just call that substrate level phosphorylation. It is a much less efficient process. This diagram just shows that there's always an enzyme that's going to help ADP, this whole thing is showing ADP, and one phosphate to be shoved together, for example, from the pyruvate. We don't need to know the details of this other than that when ATP was made in glycolysis or that one ATP that was made in the citric acid cycle, it was not made by oxidative phosphorylation. It was not made by using electron transport chain and the whole spinning of the ATP synthase. Okay, another really important thing that we need to mention here as an addendum is what happens when there is not oxygen available or not enough oxygen available. Okay. When that happens, there are two possibilities. Anaerobic respiration, which means getting energy from your food molecules without oxygen. And this can happen not so much in our cells, but in many prokaryotes, they will use sulfur, nitrate, carbon dioxide, or even inorganic metals to pull electrons from a electron transport chain that they have that is similar to ours, but different. Remember, prokaryotes do not have mitochondria. So they just rely on a different process that uses enzymes, proteins that are on their plasma membrane. But because these other sulfur, nitrate, carbon dioxide, or inorganic metals, they're not as electron negative as oxygen, they can't quite pull, there can't be as much energy pulled out of the food that they eat before the electrons that are left over have to bind to one of these other electron acceptors. Okay, so think about the general idea of what we just learned, but when we used oxygen at the end of electron transport chain, prokaryotes often will use something else. And again, it doesn't happen in their mitochondria because they don't have it. It happens in a different place by a different process. There's also something that happens without oxygen called fermentation. And that is when organic molecules are used as final electron acceptors. There is no electron transport chain at all or anything even similar to that of any kind. Fermentation is a term you have heard. You know that fermentation is necessary to make fermented foods like beer, sourdough bread, and when you ferment sauerkraut or kimchi. How does that happen? Organic molecules are reduced. Remember, gain electrons is reduction in order to regenerate NAD+. Let me show you how this works. And there are two methods for fermentation. Ethanol fermentation. Remember, ethanol is the alcohol that people drink. So we can also call it alcohol fermentation. And lactic acid fermentation. The first occurs in yeasts and some other organisms when they do not have oxygen. This is why we use yeast to create beer and wine, which is because alcohol is made. Lactic acid fermentation is a slightly different process, and it happens in the animal cell muscles. So let me show you how they work. Okay. Let's look at alcohol fermentation first. Yeasts, in general, are fungi. So they are eukaryotic organisms. They have mitochondria, and they can do the complex process that I've shown you the same exact way that we can do it, the whole electron transport chain and ATP synthase and everything. And they do use it. However, when they don't have oxygen available, because they're shut off in a barrel with grape juice, they can get by with using a much less efficient process that doesn't require oxygen. Because they're really small, they can get by with that for a while. So what happens is glycolysis, right? Glucose, and it gets broken apart to make 2 pyruvates. We have net 2 ATPs made. Remember, this is just process of glycolysis. Exactly as we learned it, no changes. During glycolysis, as you recall, we loaded 2 NAD pluses with electrons, making them NADHs. If oxygen is available, those NADHs will go where? Yes, to mitochondria, to the electron transport chain, where they will dump those electrons. But if oxygen is not available, they cannot do that, and they will not go and dump them. So then you have this accumulation of NADHs, and all of your NAD pluses will become NADHs. Like, they'll all get loaded up with max electrons, and then, like, what do you do with them? We need to get rid of those electrons that were loaded in some way so that we can have more NAD plus, which can grab this. Because glycolysis, just by itself, it does produce 2 ATPs, but that process can only work if we can take the broken off electrons and put them somewhere. If you can't do that, if there's nowhere to dump those electrons, then even the 2 ATPs can't be made because it will block the whole process. So for yeast, when oxygen is not available, it is relying only on glycolysis to create enough ATP to survive. Glycolysis can only be moving forward if electrons can be dumped somewhere onto NAD+, making it NADH. So we need to have available NAD+, which means that NADHs that have been made need to lose electrons so they can become NAD+. And the way they do it is they put those electrons onto a molecule that already exists in your cell called acetaldehyde. And when you dump electrons on them, it makes ethanol. So ethanol is just a waste product of having to get rid of the electrons we broke off during glycolysis. It is not useful for energy in the future at all. It's just like we need to get rid of this so we can at least make 2 ATPs per glucose. Which is why when you ferment yeast with, let's say for wine, it's done with grape juice, or it can be made with barley or another grain. If you're fermenting beer, you will create ethanol, which is drinking alcohol in that process, and you will make carbon dioxide bubbles, little carbonation during this process. Okay. In animal muscle cells, sometimes when you're using your muscle cells a lot and you're let's say you're running or swimming or hiking really, really hard, and you're breathing as hard as you can to get as much oxygen as possible, but your muscle cells still need to make more ATP, then there is oxygen available for electron transport chain. So you're already maximizing the speed of the electron transport chain, but you still need more ATPs. So the way that your body can do that is, in addition to already doing electron transport chain and all that to the max, it can also break up some glucose and just go through the glycolysis process, because that does not require oxygen. When you are exercising really hard, you are limited by how fast you can take oxygen and move it to your mitochondria. If you're doing that too slow for your body, then you will feel that feeling of I can't quite breathe because of this hard exercise I'm doing. So basically, you have to do the exact same thing. We need to make as much ATP as possible during glycolysis. We load them on ADH, but let's say we already have too many NADHs. All of the electron transport chains are already saturated with the electrons. So the extra NADHs need to get rid of their electrons so that NAD plus can be regenerated to help with glycolysis again. And that happens by putting them directly onto pyruvate. So you make pyruvates and then you put more extra electrons on them in order to regenerate NAD plus. That waste product is lactate, which is another way to say lactic acid, which is why we call this process lactic acid fermentation. Okay, so these are the two. Fermentation are two versions of glycolysis that happen when products of glycolysis cannot continue on to pyruvate oxidation and all the other steps because no oxygen is available or not enough oxygen is available. Okay, fermentation is just to regenerate NAD plus, get rid of those electrons that we can't use up so we can at least get ATPs made through glycolysis. Glycolysis is not very efficient. Per 1 glucose, you only make 2 ATPs, net 2 ATPs, but it's better than nothing. And this process, glycolysis, in all of its versions, is the oldest way for cells to create ATP energy, evolutionarily speaking. And that's probably why glycolysis even exists in our cells now when it's clearly not efficient, can only make 2 ATPs. It's a whole evolutionary building step. How do we get ATP made or energy from catabolism of proteins? So how do we get energy from the breaking up of protein? Let me show you in brief. First process is the proteins get into your stomach and into the small intestine, and they get broken up into individual amino acids. The first step is deamination. The amino group from amino acid has to be removed, and it's taken away as waste product. And the remainder of the amino acid is converted into one of the intermediates of glycolysis or the Krebs cycle. It's actually one of the intermediates in the citric acid cycle. And if you scroll back, I was trying to find the number of the slide that it was. And if you scroll back to slide number 23, you can see that alpha ketoglutarase is just one of the intermediary in between molecules of the process. You do not need to remember that. You just need to remember the basics, that to use amino acids as energy, we have to deaminate them, and then the remainder of the molecule will get plugged in as an intermediate in the whole long process. The same thing happens with fats. Fats are broken down to fatty acids. So you take the long chains of hydrocarbons and you break them into little pieces. And then those little pieces are inserted into same thing into the process. This is an oxygen dependent process. So if you want to burn fats, that has to be done in the presence of oxygen. So it has to be done during aerobic activity. It cannot be done anaerobically. Okay. So in this case, take a fatty acid, it gets broken apart, and the little 2 carbon pieces are going to be acetyls. So the acetyl will just come into the Krebs cycle, just like all other acetyls, which is how fats are catabolized. They're just broken, the long hydrocarbon chains are broken down into 2 carbon pieces, which are acetyl molecules, and then acetyl will get shuttled into the Krebs cycle by coenzyme A, just like acetyl that came from glucose. So all in all, there's a summary of macromolecule degradation or catabolism, how we break it down. In general, if nucleic acids in your body are broken down for energy, it means you're going to die in just hours or even less, because those nucleic acids are there as code. And if you have to break down your code and your blueprint for how to live just to get energy, then that is the end. We evolved to mostly use polysaccharides or monosaccharides as energy. Lipids and fats are also good energy sources. Proteins are not meant to be as energy per se, but they can be used for energy when necessary. And of course, nucleic acid is just like ditch last effort. That's not going to get you very far. Okay. So that's gonna be it for chapter 7.